couple of seconds, and then I'll take it away, and we'll crack on.
Are all the Q&A experts on the West Coast? Just out of curiosity, I know Gene, you must be in San Diego now. Oh, looks
Yeah,
like good
yeah.
morning.
We just sort of roll out of bed, so.
So I'm not the only one who hasn't had a coffee yet.
Well, I'll keep it light for you, gentlemen.
Yeah.
So, yes. Well, in that case, good afternoon from the European side of things. Good morning to our US counterparts who are tuning in for the live Q&A session with Ask the Experts. I'm joined by Gene, Oliver, and Gu Cheng, who have taken the time out of their day to join us to present and also answer your questions from the session. Oliver, we'll get through this quickly so you can have your coffee. I hope that's okay. And thank you for all of you for joining us. Oliver, I'm actually going to start with you if that's okay on the question front. The question for you is, what is the maximum number of biomarkers that one can detect with the Codex technology today, and is there an upper limit?
So the maximum number of biomarkers that one can detect with the Codex technology today has not been empirically measured as far as I'm concerned. So we do not know, we are not aware what the upper limit is. So in theory, you can do 100, 200, 300, 400, and so on. The maximum number of reagent barcodes that are currently available to detect biomarkers that you can buy commercially at today as an inventory is 43, I believe. So if you were going to do this experiment today with our product, you can detect 43 biomarkers. And that is really, again, that is just because that is how much reagent we have to do this experiment. However, theoretically speaking or experimentally speaking, I'm not sure what the maximum number is. I don't know. I don't think there is one. The only limitation I would see is the steric availability of antigen binding sites on proteins that could set a cap on this somewhere, but we are unaware of it. And I would suspect hundreds. So that will be my answer for you.
Fantastic. I appreciate it, Oliver. Thank you. The next question is for Guo Cheng. I am a biologist who does not have any programming experience. Is there an easy way for me to use Gitto to analyze my own data?
Sure. We try to make this as user-friendly as possible to biologists, and we have a number of ways to make this easier. And one of the things that we do is that we offer the examples for different data sets, and so you can just follow the instructions in these examples, and then to pick the steps that you want. We also post a number of the how-tos. Go to this specific functions if you do how to do the clustering, how to find the spatial patterns, and how to do the interactions, and so on. We posted a YouTube video, so it's kind of more interactive way to guide. So that's all of that can be found in the Jota website. So lastly, so this is what we have done so far. Something that we are currently doing is that we are making a binder-based notebook, the Jupyter notebook, so that you can play with the data yourself without actually installing anything. And lastly, that's one thing that we are going to do in the in the future. We're going to start doing this. Future is going to offer like tutorial sessions, educational sessions, there's various conferences so that we can interact with the users directly that way. So these are some of the things that we're doing to make it user friendly.
Fantastic. Thank you for sharing. And just before I ask a question to Gene Yeo, I just want to remind that everyone joining us on the session, if you do have any questions during the session, please put them into the messaging bot. Our production team will pick them up and share them with me to ask directly. So my question to you, Gene, is with spatial biology in general, it's evolving at a rapid pace. So in your opinion, how does the future look like for this area in your opinion? And I'm happy to have Oliver and Guo Cheng join in afterwards if you'd like.
I mean, I can speak so broadly. There are many experts in this area. I'm for my lab, we're particularly interested in combining orthogonal technologies, spatial technologies, biochemical fractionation, but really looking at disease models where we can perturb the system and evaluate how subcellular RNA or gene expression is altered, right? And then in consequence to the perturbation as a way to model diseases. And so I would imagine that we will sort of slowly move away from just characterizing, fixed tissue or samples from patients, but then move to disease modeling as well, and using using sort of spatial information as just another metric to evaluate what is the delta between disease and normal, and then once we have a clear metric there, then eventually move that into a miniaturized format for high throughput screens. Yeah.
Fantastic. Appreciate that. Thank you. And sorry for putting you on the spot a little bit there, Gene. Oliver,
No,
do you have what's exciting you about the spatial biology space, if you don't mind commenting on that? What are you looking forward to most in the coming years?
I guess that's a good question. I think the technology has had a profound impact already on the just not the life sciences, but also the clinical and biomedical community. And I think Gene has sort of alluded to that when he says disease modeling, and I agree with that. I guess from our perspective, it's really what we're looking forward to is the advanced understanding that spatial biology will bring in the clinical sector. I think it's a no-brainer, or it goes without saying right now that the value of this technology, the information content that these experiments bring nowadays, they stand to have a transformative effect on the way that we diagnose disease and hence better understand diseases. So what I'm really looking forward to that is for us to, embrace these technology in a clinic and to finally, really embark on new diagnostic approaches that will hopefully end up, significantly bettering our ability to detect and treat disease.
Fantastic. I appreciate your input on that. Gene or Guocheng, do you want to add anything to that? Something may have popped up before I come back to the questions. I'm interested to see if there's anything additional you'd like to add to that, because Oliver's highlighted a great point there. The actual work of spatial biology going into the clinic has been a hot topic over the last few days. It's been mentioned quite a few times. So if anyone has anything to add, it'd be great to hear.
Yeah, I think the bottleneck there, it's not a technical bottleneck, but an adoption and in some sense, a regulatory bottleneck, right? Because one would argue let's say, for example, as pathologists, I'm not a pathologist, but as colleagues that I have as pathologists, you're sort of asking them, here's a method where instead of H&E stains, which one would argue is like a black and white television set, here's a high definition HDTV equivalent, right? So are you comfortable with digesting the data, or do you need it, and so on and so on? And typically we've heard from our clinical pathology friends, there's like, well, this is a bit complicated, right? And regulatory will have issues with it, right? So I think it's not a technical problem. I think we can get there, right? I think it's a regulatory issue. I'm not sure. I wonder if Oliver and Guocheng have comments about that. Yeah.
I think that's going to require a cultural shift as much as a technological. I think this is an outstanding point. We still look at monochromatic H&E stains. So I think you're spot on what you're saying.
Yeah, I totally agree as well. I think that's there's going to be a lot of issues that we have not be used to prior to the spatial area. Just as a data analyst point of view, so one thing that I think I'm really excited about this field is that there is before we actually enter this area, there's a very little consideration or thought about how these individual cells, they are really organized together, how they interact and how they form the structures and all of that. I think the people that in the past that have been used to the way of looking at things very intuitively, right? So they say, here's this, here's that, and I tend to make conclusions right there. But moving forward, you have to be more cognitive, more rigorous, and systematic, and so on. I think we are still really lacking that kind of fundamental conceptual level of tools or views or whatever.
Okay, so yeah, you've all highlighted some different sort of challenges to potentially overcoming in the coming years. One of what Gene shared in terms of obviously a cultural shift of actual human behavior of using new information that's provided, and also obviously the FDA side of things. Will these things be approved? Do you guys do you foresee any potential other challenges that we may need to overcome in this space in the coming years?
In terms of regulatory, you mean?
It just in pretty broad terms, in terms of what other challenges do you think that may arise during the next few years in this space?
Well, so I think that the immediate challenge would be that people would have so many technologies to choose from. they are a little bit confused at the moment, because I work at with some of the biologists that are in our or clinicians. They say, okay, so we know all these fantastic technologies, but which one should we get? And if we use some technologies developed now, would they going to be changed in 2 years from now? I mean, for them, it's kind of like a investment issue. In addition to this kind of regulatory issues, I think that's I don't know how, with the field moving so fast, I don't know how Gene and other people will think about it. So,
I mean, it kind of reminds me of the. I mean, it kind of reminds me of the microarray days for gene expression measurements, right? Where at one point there were new array technologies that appear on the scene every like 6 months or so, but then it became clear, that you needed to have, in some sense, the responsibility of in terms of the academic labs that churns through the different technologies, and at the end, I think it was technologies that were highly reproducible, where there were bioinformatic pipelines that could easily crunch the data sets on those, and where the price point was like decent enough, where academic labs could afford them, and that they could become useful for clinical use. But and so that took like almost 10 plus years, right, before that ended up being one or two companies that existed. So it may be that we are seeing the same froth, and maybe in 5 years you see one or two major platforms, right? But another lesson from that era was that I'm not sure if array technologies at the end became useful as clinical tools, right? Because once you found interesting gene expression differences, just for the ease and also convenience, ease, regulatory requirements to get to a limited set of targets, that then always boiled down to qRT-PCR as what you use for a clinical test, right? And so I'm not sure if the arrays actually ended up moving into that realm. It was good at discovery tools. And my guess is that if we watch carefully in the spatial, biology area, I think all the tools are fantastic discovery tools. And it may be that in the next 5 to 10 years, one would argue then those will end up becoming, for clinical diagnostic, right, simpler and simpler, subsets of these, targets, right, cleaner and cleaner pipelines, and almost to a push button solution for clinicians, right, pathologists to look at. So that's sort of, looking into a very fuzzy crystal ball. This is what I'm guessing will happen.
Fantastic. I appreciate that, Gene. We've actually had a question come in during the session for Oliver. I think my prompt might have got someone to notice this. So the first comment is, Oliver, I enjoyed your talk. I was curious about how do you decide to pick a probe for a cell type that is only 0.07%? Was it scRNA-seq clusters?
That's a great question. So this is, let's call it discovery biology. So I'm not an oncologist or a breast tissue biologist by any way, shape, or form. I'm actually a neurobiologist by training. So when we did this experiment, we picked a menu of probes that we wanted to characterize epithelia in the breast tissue. That was a very simple hypothesis. That was our premise. We want to use probes for so-called basal cells and probes for so-called luminal cells. And the idea was to get as much coverage of these different epithelial cells as possible so that we could then eventually build a catalog of what these epithelia look like in different conditions. And the idea being that, for example, a breast cancer would have different type of epithelium than a normal tissue. That was our premise. We didn't know what to expect. Then we ran the data analysis and we recognized this small cell population of 0.04%. And initially, actually, my recommendation was to dismiss it as noise. I was like, what is this? We don't need to worry about this. It's just such a minuscule cell population. We don't care. It's only after that we went and found that these data have been published elsewhere. And then we aligned with now a very active collaborator of mine to do further validation studies to see that these cells actually exist. So this is purely done out of accident, but I think it's a great example of how these technologies, and I think Gene said the word discovery several times over. Yeah, that was just an accidental discovery, if you could say that. I did not have any intent to do that. It just the data presented themselves onto me. I'm much more of a classical, let's say, anatomist than I am a big data user. So, and then I see this, I'm like, what is this? It's my gut feeling was it's noise, but it turns out it wasn't.
Yeah.
It was pretty cool.
Fantastic, wonderful. That's great. I've got a question for Gu Cheng. I am a method developer. Is there a way I can get my method implemented in Giotto?
Yeah, so we have a lot of ways to help others to do the integration. So we have at the website, we have a contributor page. So we have some instructions. How do you format your codes? How do you, what's the style? How do you convert your data structure to what's the Giotto-like structures? And we have some examples. We give an example as to show you if you have a tool, then how do you actually, implement that. And I think that's we are in the future. We are really open to collaborations with other method developers, and we're very happy to interact with you if you want. And we also want to make the easier for people who use other common software such as Sora, such as ScanPy, and so on. They can navigate easily between the Giotto and the other, the toolboxes, so they can use the strengths of all of these tools together. These are some of the things that we are considering doing this, make it friendly to the method developers.
Excellent. Thank you for sharing. We have around about 3 minutes left before the session comes to end. And normally I end on a pretty open question about what to expect in the space in the coming years, but we've already covered that. So I'd like to do something different if that's okay with you. I'd like to ask each of you what you're currently working on. So Gene, what you're currently working on at the University of California in San Diego Health. Oliver, what you're working on at Akoya and what to expect, just to essentially, in a way, give yourself an opportunity to potentially plug your research, where to connect with you and what to look out for in the future. Same with you Cheng. So Gene, if you'd like to go first, take it away.
Sure. Yeah, so my lab works on RNA biology, right? And so we look at spatial biology as a technology, as an opportunity to not just do identification of cell types, right? But actually to understand how RNAs are actually moving and in very discrete packages in the cell with relevance to diseases like neurodegeneration, where there's RNA transport issues, and other in other organs in future, but we're mainly focusing on neuroscience today. And our goal has always been to move some of these ideas into our RNA therapeutics realm. So these are for us technologies to understand sort of RNA biology. Yeah.
Fantastic. Oliver, what are you currently working on at Akoya, and what can we expect from you in the future?
So at Akoya, at the moment, we work at protein biology and protein spatial biology. We have there's two ways that I look at this. One is the number of analytes that we look at. So we look at a lot of analytes, or the number of tissues that we can look at. So what we're working on is sort of an interplay between really high throughput, like looking at 100 tissues a day with a lower level of analytes, versus looking at 100 analytes a day, only look at one tissue a day. So one of the things that I think is a really interesting question is, think about the data. if you do 100 tissues a day with 5 analytes versus 1 tissue a day with 100 analytes, in some sense they meet. At the end of the day, you still have just 1 million bytes each. How do you really, let's say, massage these data in order to find patterns, trends that actually matter? So, and this is especially true. One of the things that we're working on, that I'm working on, is spatial biology is cool. It's exciting, and everyone's discovering everything. We just talked about this little cell type that I found. What does it actually mean? So this idea of getting to a high throughput stage and understanding what these patterns mean on a larger scale is something that I find that tantalizing, and something I work on right now, and I will continue to work on.
Fantastic. Thank you for sharing, Oliver. And to end, Guocheng, what are you currently working on, and what can we expect from you in the future?
So the immediate thing that we're working on is to work with several disease experts to apply this to situations like neurological diseases, cancer, immunological diseases, and to get biological findings. But we also, as a computational biologist, we really like to push the limit of the methods, tools, development, and so on. And one of the things that we are mostly interested in right now is trying to extend this not just from the spatial transcriptomics point of view, but also look at the DNA structures, the epigenomics, and to understand how the genes are regulated in the single cell, specific manner with the surrounding environment. These are the kind of things we're doing.
Fantastic. No, that's excellent. Well, we've just slightly overrun, but it's okay because it's actually technically a break. So I guess we're all right in that respect. So Gene, Oliver, and Guocheng, thank you very much for joining me on your side of the planet at very, very early in the morning. So thank you for joining us live. We appreciate you giving the answers to the questions and also highlighting what to expect in the spatial biology world. We look forward to obviously continuing to witness your work, and we hope you enjoy the rest of the conference and